Namaste students, I hope you all are fine. So today we will discuss about the topic transcription of molecular biology of inheritance. Yesterday we talked about the structural gene, which is also known as the transcriptional unit, and it contains the promoter as well as the terminator region. And we discussed that when RNA polymerase it binds with the promoter region, then the transcription it starts, and when it reach at the terminator region, then transcription it stops. Means the RNA which produced, then it will get separate from the DNA helix. Now today we will discuss this transcription process in detail. So students, next we will discuss about the transcription in prokaryotes. Transcription in prokaryotes, it occurs in the cytoplasm region. And what are the requirements for the transcription in prokaryotes? It requires the RNA polymerase enzyme. It is also known as DNA dependent RNA polymerase. And second, that is sigma subunit for the initiation, and the rho factors for the termination, and NTPs. What are NTPs? These are the nucleotides, means those nucleotides which will add by the action of the RNA polymerase. Means when the RNA polymerase it move over the structural gene, then the NTPs they will add on, and then it helps in the synthesis of the RNA chain from the template strand. And in case of RNA chain, like in the DNA, thymine that is present. So during the formation of the RNA chain, uracil it will be present in place of the thymine. Now next we will discuss the transcription process in case of prokaryotes in detail. So there are three processes. First, that is initiation. Second, that is elongation. And third, that is termination. So as you can see in this diagram, that when the RNA polymerase, it will bind with the promoter region, and where the promoter region it is present at the 5' end of the structural gene of the transcriptional unit. So when the RNA polymerase it binds with the promoter region, then this RNA polymerase it will recognizes the sigma subunit. Then it recognizes the sigma subunit, and then it will bind with the sigma subunit, and then it will move over the chain. One more thing, only one RNA polymerase it is responsible for the synthesis of all the three types of the RNAs, means the mRNA, tRNA and the rRNA. Only one RNA polymerase it requires for the synthesis of all the three types of the RNAs. So when the RNA polymerase and the sigma subunit they are present, means they will combine with each other, then together with each other they will form the transcription initiation complex. Transcription initiation complex. And then the transcription it will start from the promoter site, which is also known as the transcription start site. So as the RNA polymerase it will move over the DNA helix, then you can see here that the DNA helix it will get uncoiled. and the bases that are present over the DNA, they will also get exposed. Okay, so when the bases they will get exposed, then what happens? Then the antiPs that are added, and this process is known as elongation. It helps in the elongation process. So as the antiPs they will add on by the action of the RNA polymerase, then it helps in the elongation of the RNA chain, and when there will be the elongation of the RNA chain, and then when this RNA chain, or you can say the RNA polymerase, it will leads, means it will go to the termination sequence, then the rho factors which are present in the cytoplasm, they will bind with the terminator region, and the rho factors they are responsible for the termination process. So when the RNA polymerase it reaches the terminator sequence, okay, means the terminator region, then what will happen? The rho factors they will add, means they will join, they will bind with the terminator region, and when they will bind with the terminator region, then what will happen? The RNA chain it will get separate out from the DNA helix, and this is known as the termination. See once again, what is initiation? Means when the RNA polymerase with the sigma subunit they will bind with the promoter region, then this is known as the initiation, and this complex is known as transcription initiation complex. And what is elongation? Elongation means when by the action of the RNA polymerase the hydrogen bonds they will break down that are present between the DNA helix. So when these hydrogen bonds they will break down and the bases it will get exposed, then what will happen? The antiPs that are joined there, the antiPs they will add on, and always the synthesis it occurs from the 5' to the 3', because here the 3' to 5' and it is acting as a template strand. Now as the antiPs they will add on, and when the RNA polymerase it will reach to the terminator region, then the rho factor it will binds with this, and the RNA chain it will remove from the DNA helix. So this is all about the transcription in prokaryotes, and only one RNA polymerase it requires for the synthesis of all the three types of the RNAs, that is mRNA, tRNA and rRNA. And the transcription and the translation process they occur simultaneously. Okay, it means that the RNA chain when it will synthesizes, so at the same time the translation process it will also occur. Like in case of eukaryotes, first transcription it occurs and then translation later on it occurs, but in case of the prokaryotes both the processes it will occur simultaneously, because the transcription and translation both the processes it occurs in the cytoplasm region. But in case of eukaryotes, the transcription it occurs in the nucleus and the translation process it occurs in the cytoplasm region.